recommendation. Professor Basu, please. So now it is 10:19. So you have been allotted for 40 minutes. I believe within these 40 minutes you will be able to finish your topic. Professor Basu, sir.
Yeah, thank you, Dr. Mondal. Let me just try to connect the screen. I don't know how to do that. Yeah. Is my screen visible to everybody?
Yes, sir, it is visible.
And if I go to the next slide, is it going smoothly?
Changing. Yeah, it is visible. It is visible, sir. Yes.
Okay. So, good morning, everybody. Thank you, Dr. Mondal, for your kind introduction. I would like to thank Dr. Jui Chakraborty as well as Dr. Suman Mishra for their kind invitation. It has been always a pleasure to deliver a talk at CSIR-CGCRI. I have been in contact with several CGCRI scientists over last 1 decade or so since I came, more than 2 decades since I came back to India in 2001. So today's topic is chosen in a manner that I expect that this presentation is going to give an overview of the field. And as the field is growing in India and internationally, and I would expect this presentation also will inspire the next generation researchers in this emerging field. So this is the plan of the talk. When I say crossing the boundaries, essentially what I mean to say that biomedical science represents a truly interdisciplinary field where concepts and ideas from multiple fields converge, and the impact is significant both in terms of the country's economic impact as well as the societal impact in terms of the healthcare of the large cross section of the people in several countries. Then I would follow with this international status. I'll give more like Indian landscape in the 4th section. And most importantly, I'll provide a set of recommendations which I have written in much more details in my recent book, which is published by Indian National Science Academy together with Springer Nature. And that will be, then I'll close my presentation. Now, for many of the young students, these biomaterials that often in various interviews and other places, if I ask that people that, how can you define biomaterial, people often stumble to give a very acceptable definition. So biomaterial is a class of materials with the proven compatibility with the components of the living system, means protein, blood, cells, and tissues. This is the introductory slide. Often I show it in many of my biomaterial centric presentation. So what you see in the cartoon is that eukaryotic cell, which is a truly nucleated cell, which is adhering to the biomaterial substrate through focal adhesion complexes, which are nothing but the cluster of the cell surface receptors and then adds up proteins at the biomaterial surface. And what you see, the string-like structures within the cell, so they are essentially actin filaments. So essentially it reflects that if the cell will attach and adhere and proliferate on the biomaterial substrate, it must be associated with the cytoskeletal reorganization. Now, the property which distinguishes biomaterials from other classes of materials is the biocompatibility property. And this is a holistic property which renders the biomaterial with most appropriate beneficial cellular or tissue response for targeted clinical applications. So what you see in the cartoon that, that when a material is placed, in the in vivo system, whether it is an animal model or human system, then first thing is what would happen, that protein gets absorbed on the biomaterial substrate. That will be followed by the different type of cells which will be getting attracted to the biomaterial substrate. Now these cells will in turn will send out the cell signaling molecules or cell signaling proteins to send the information that this particular material is biocompatible, so that other cell types also will come and adhere on the biomaterial substrate. Ultimately, all these activities will lead to the formation of foreign body plant cells, and then they will subsequently synthesize the collagen, and then they will form extracellular matrix around the biomaterial implants. And then net result for any synthetic implant which is not bioactive or which is not bioresorbable is the formation of collagenous bag, acellular bag, which will kind of shield the biomaterial from other parts of the osseous system. So this happens, antibiotics tell you to come back to them after 3 to 4 weeks, because that is the scientific reason for this particular timeline that is often prescribed. As I said in the introductory slide, represent truly. So essentially, this brings the concepts together from these two disconnected disciplines, which is very important to mention, because material science and biological science, there is nothing much to common. If you say physics and material science, there is a lot of commonalities. If you say chemistry and material science, there is a lot of commonalities. But that much commonalities is certainly not there between these two fields, material science and biological sciences. Now, what you see in the periphery is the biomechanics, manufacturing science. So these are important for the for the development of different implantable biomaterials. Then comes toxicological and veterinary science. So toxicology science brings the expertise of the in vitro studies, particularly genotoxicity studies and so on. Whereas veterinary science is important for the preclinical studies. Now, ultimately, the medical science gives you input to define that what are the unmet clinical needs or what are the clinical challenges, and the impact of this biomaterial science will certainly be towards the healthcare. So this brings me to the next slide. So what are the spectrum of applications of the biomaterials? Now, what is popularly known as a phrase called head to toe. So that is the biomaterials application expand from head to toe of a human being. Now, if we start from the top is a bone flap for cranioplasty. I'm going to show you one example from our own clinical study. Then comes cochlear implant. Now there is all kinds of these musculoskeletal joint applications, like whether it's the shoulder prosthesis, total hip joint replacement, total knee joint replacements, lumbar disc replacements, and so on. Then comes more critical applications like cardiovascular implants for the stents. Then urological implants. Again, we are also working on the urological implant, particularly polymeric biomaterials. Now, one of the key things that is important for us to recognize that more than 75%, or I would say around 80% of the implants which are used in Indian hospitals and clinics, they are all imported. Now, these implants are essentially fabricated in the Western countries, supplied by the Western companies, which are essentially suited for Caucasian populations or the Western populations, and many times they do not have exact anatomical fits to the Indian demographics. So there is indeed a need for high quality indigenous implants, which would be most suitable for Indian population. Now, what you see in the bottom concept triangle is that engineering and physical sciences, their concepts lead to the development of the implantable biomaterials. But the medical and biological sciences, they give us an understanding of the living system. Now, their interactions, that how implantable biomaterials, when placed in a living system, how they interact with the components of the living system, that is the central theme of the field of the biomaterial science. Now, this biomaterial science tetrahedron has been drawn based on the similar material science tetrahedron, which you often see on some of the famous journal in the field called Acta Materialia. So what you see in the biometrics development in one corner, then goes to the biocompatibility evaluation, meaning that more is in vitro and in vivo, then goes to clinical studies in the human patients, and goes to the public health. So ultimately, as I said, that biometrics has to benefit the public health care. And it is essentially public health care, the clinical needs will drive the development of new biomaterials. And it is also important clinical translation. So any study which ends up often with the in vitro study, that may not be of much use for the society unless you take it to all through these preclinical studies and up to the clinical studies. I'll show you some data which is taken from different market studies, just to give you an idea that what is the current market need. Now, if you look at this particular bar plot, what you see is that global medical devices market is increasing at the compound annual growth rate currently at 7.5%, whereas that market in India, it is growing with the compound annual growth rate of 23.2%. And if you look at that numbers now, it is essentially $5 billion in 2016, and it is projected to reach somewhere close to $20 billion by the end of 2024. Now, if you look at the two particular application areas, one is dental and one is orthopedic, what you see here that both of them, they will constitute close to around $3 billion, particularly at the end of the 2024. Now, the important aspect often people ignore that India attracts a lot of medical tourism, and these medical tourists come from our neighboring countries like Sri Lanka, Nepal, Bangladesh, Bhutan, and so on. And at the same time, India attracts a lot of medical tourists from African nations. Now, there is a specific cost advantage this particular slide essentially tells you. So if a patient goes to United States and compared rather than going to India, that if a patient goes to United States, he or she is going to spend 7.5% more cost for the identical medical treatment for the same treatment compared to India. So certainly, as you see that affordable health care attracts medical tourists in India, and it is certainly at a higher level compared to many of the Asian as well as other countries around the world. Now, having said that, the funding in this particular area is not significantly high when I have recently compared with the funding that is being pumped in countries like UK, United States, and Australia, for example. Now, research funding in this area is essentially sponsored by our national funding agencies, DST, DBT, and DST started this. CRB has started this imprint program, and CRB also has health sciences program, and DBT has two programs. One is the bioengineering, one is the biodesign program. And then there are private funding agencies like Bill and Melinda Gates Foundation through BIRAC. They also support some kind of biomaterials which can be directly gone for healthcare applications. And if you see this funding in terms of the million INR, and when 100 million INR is equivalent to $1.3 million, you see that this data is incomplete. It is essentially in 2018, 2019, but that 27 and 2018, if you see, it is almost like 200 million INR was funded through various sponsored projects in India. Now, international status, this slide essentially gives you the overview of the significant activities that is going on and some of the most representative groups around the world. If you start with the North America, Christy Ansels Group certainly is doing excellent work in the field of hydrogels for various biomedical applications. Then David Kaplan, who is the editor-in-chief of ACS Biometrics Science and Engineering, he's globally known for his work on silk Bob Langer at MIT in United States, I think he is the godfather in the field of biomaterials, and I always tell many people that if somebody is going to get Nobel Prize, I think Bob Langer is going to get the first one. He's become really billionaire now with this Moderna vaccine that has come to market, and that is the brainchild from Bob Langer's company in Harvard, MIT. Cato Lawrenson is one of the world's most famous African-American scientists, and who is created recently history, being the first surgeon to become elected as a fellow of all National Academy of Engineering, Science, and Medicine in the United States. In Europe, there are several groups. In UK, Serena-based groups in Cambridge, who is to have a UKD project with NML Jamshedpur. I think Dr. Suman Mishra may be knowing her. Then comes this Mark Werner from Switzerland, Aldo Boccaccini's group. Aldo Boccaccini is well known in CGCRI community because of his connections with the glass community around the world. Asia Pacific, in India, IITs, ISC, and so on. But some of the groups which is worth mentioning is Dietmar Hartmaker group at meeting has received worldwide attention from Queensland University of Technology. Then comes Tabata's group in the field of drug delivery from Kyoto University in Kyoto, Japan. Now, those are more like individual groups, but there are also multi-institutional research platforms. Like, for example, one of them is Prometheus Group in Catholic University Leuven, Belgium, my alma mater. And this is one of the group that I have seen which is physically located within a hospital. So they have a university hospital, and this group is conceived, nucleated, and functioning within the university hospital complex. Abhay Pandit's group, Abhay Pandit has started this QRAM, Center for Research in Medical Devices, Galway, Ireland. Abhay also comes to India very often, and then he has right. So Harvard University has a Wyss Institute, and Wyss Institute has a unique translational ecosystem. They essentially support many entrepreneurs on campus, and their task is to take this their task is to exploit the innovation ecosystem in academia, then goes through concept validation, technology refinement, and goes to commercialization. This is another one that has come up in a big way in UK. This is National Materials Research Institute, and which is funded by Henry Royce family, and that's why it is known as Henry Royce Institute at University of Manchester. And they have one of the vertical is biomaterials, and particularly bioelectronics. So they have these bioelectronics, smart materials and devices, and they have additive manufacturing suits and 3D tissues, cultures, and so on. So essentially, the aim of this institute to act as a hub to cater and to accelerate the biomaterials research in entire UK by leveraging their contacts with the multiple academic institutes like University of Sheffield, University of Cambridge, and so on. But this hub is located at University of Manchester. Now, coming to India. Now, India is there is significant activity certainly is going on, which is quite impressive. And depending on north, south, east, west, I have tried to put together as many groups as I know, which are actively doing. But one disclaimer is that certainly it may be possible that some of the groups are missing. That is clearly unintentional. In the northern part of India, Devrupal Nahid's group at IIT Roorkee, now Naresh Bhatnagar's group at IIT Delhi, and sure, of course, they do wonderful work in 3D bioprinting. Then comes to the western part in VNIT Nagpur and IIT Bombay. They have called Betik Center, B. Ravi's group, and also Rinti Banerjee's group is working on tissue engineering in IIT Bombay. In eastern part, Kanagra's group at IIT Guwahati, CSIR-CGCRI certainly has done some of the outstanding work. Then Arvind Sinha's group at NML Jamshedpur. In the southern part of the country, there are national labs as well as some of the institutes, NIT, and as well as one of the most prominent institute, and which is also known as the National that is Sri Chitta Kiruna Institute of Medical Science and Technology, and that is based in Trivandrum. So one of their landmark product is the Chitra heart valve, which has been marketed very extensively in India and many other countries. So as mentioned in the introduction, while introducing me at this talk, that we are currently leading one of the India's one of the translational center of excellence. So there are 3 unique products, both 2 are for orthopedic and 1 for dental. One is a ceramic femoral head, polymer ceramic acetable liner. This is currently being tested using hip simulator at CSIR-CGCRI. So these are some of the statistics in terms of total number of publications and so on, and this technology is being transferred. So just to give you one example of this product which is come out from the center, the HDP UHNWP hybrid nanocomposites, where very small amount of graphene oxide has been added, because we have to be careful in terms of the carbonaceous reinforcement, because it should not cause toxicity to the materials. So what is the strategy we have done? That we have used the blend of the HDP UHNWP, because if you want to add a lot of reinforcement to HNWP per se, it is very difficult because of the high viscosity. And we have chemically grafted this graphene oxide, then added to HDP UHNWP blend in order to increase the strength to the level of 65 megapascal, which is quite impressive as far as the strength of this materials is concerned. Then we have done a series of the in vitro study using the different cell lines like osteoblasts, as well as the fibroblasts and stem cells and so on. We have done some of the hip simulator study as well as spin on D-square study, then confirmed that confirmed the absence of any cytotoxic wear particles. Recently, we have completed blood compatibility. It essentially shows less than 5% hemolysis, which is acceptable. Osseointegration study was done quite before on different kind of compositions, and that was done both at All India Medical Science and Sri Chitra for up to 26 weeks longer time. Now, what is the new thing we are currently adding to this particular research theme is that we are trying to connect the machine learning algorithms to biomechanical analysis in an effort to accelerate the different design of the acetabular liner for faster integration with the pelvic bone. And in this particular case, we are using this bone conditions, subject weight, and implant design as the input variables, and the output variables are essentially von Mises stress and von Mises strain. Now, whatever machine learning we have done so far, it shows fairly close correlation between the predicted value from machine learning with that of that finite element analysis value. And this essentially cartoon shows that two phase in the gate cycle, not the complete gate cycle. One is a loading response and one is the mid swing response. Dental implants, so TI6L4V or titanium has been conventionally machined to a particular dental implant design, and these dental implant designs are essentially quite unique, and this is being patented. We have done the biomechanical analysis just to show you that what is the von Mises stress contour around the implant. Then we have done the insertion and then removal torque, both in human bone as well as the pig bone, in addition to the synthetic saw bone. And what it shows that with respect to the control implant, our implant, ISC implant, shows much less insertion torque in case of much less insertion torque, both insertion torque and removal torque. Subsequently, we have done dynamic wear study using dental wear simulator, and then we have implanted these implants in the rabbit condyle, and this essentially shows you that how this micro CT of this implant after the implantation, and then essentially it shows the neobone formation histology images, and then we are currently taking this pilot scale manufactured in the MM Private Limited. This is one of the work at NIT Surathkal with Somen Mondal's group. So, inspired by this so much natural resources at Arabian seashore, I've taken this slide essentially shows one of those marine resources, cuttlefish bone, and cuttlefish bone derived, hydroxyapatite, essentially calcium carbonate, then it goes to calcium oxide, then you treat it, then you make this hydroxyapatite. We have done this scaffold, hydroxyapatite scaffold. So one of the important things that we have established that, in the marine derived bioceramics, now if you make this scaffold with different porosity, how the strength and elastic modulus they vary with porosity, and it shows kind of little bit non-linear type of relationship. And one of the unique things of the marine-derived bioserum is that it supports the biomineralization in terms of the calcium phosphate crystal nucleation and growth very rapidly within 3 days of the incubation in the simulated body fluid. This is the overview of our work on 3D printing and 3D bioprinting of implantable biomimetic scaffolds. So BinderJet 3D printing allows one to print these biomaterials at room temperature, at close to physiological temperature. But one of the key things is to develop an understanding that when the binder interacts with this powder bed, how the binder infiltrates into the powder bed, and there various theoretical models like Washburn model was used and adapted to predict this time of infiltration. And based on this time of infiltration, we have essentially tuned the 3D printing parameters. So we have taken several materials, but this particular slide, the left-hand side, it shows as vanadium, because this is one of the FDA-approved material, and it shows the strength reliability as far as the viable plot is concerned around 8. So 8, but the strength reliability in terms of M parameter is 8 in this kind of highly porous microstructure with 98.4% interconnected porosity is quite an impressive number. Now, in terms of the cytocompatibility, it shows very good cell proliferation, and it's certainly better than commercial TI6L4V, and that is expected because of the porosity in the structure. To take this work forward, we have done that exosynchrotron study at Diamond facility in UK, and that allows us to quantitatively analyze this infiltration kinetics, both in space as well as in the time front. And we also extended this Washburn theory, particularly to incorporate Denusk and Holman model to find out what is the wetting volume and how it increases with time of interactions. Now, based on our experiences of the printing of biomaterials and cells, we recently started more than 2 years ago work on 3D bioprinting. We have already developed this. We are part of this design and manufacturing, which was essentially done at Tivasta, this 3D extrusion bioprinter. We are trying to reduce the weight of the 3D bioprinter so that it can be used in the India's futuristic space bioengineering program. So far we have done bone scaffold with gelatin methacrylate as well as nano hydroxyapatite. This shows that what is the typical scaffold architecture and dimension we can print. Now, in terms of nerve regeneration, particularly peripheral nerve regeneration, we use the photopolymerizable cross-linked bioink, that is gelatin methacrylate. And the preclinical is under progress in the rat model. So essentially, we put this hybrid conduit in the sciatic nerve when it is cut, and then we place it and see that how fast this nerve is regenerated at that place. And this is one of the work that I have started in IIT Kanpur long time back in 2006, and we still continue in our group. That is bioelectronic medicine, essentially programming stem cells for regenerative engineering. So the left-hand cartoon essentially shows the concept. The concept is that if you grow stem cells on a bioactive or on an engineered biomaterial substrate, and you give this electric field stimulation, so this stem cell can undergo either osteogenesis to a bone cell or cardiomyogenesis to cardiac cells or neural cells or neurogenesis. And in all these cases, you have to tune the substrate conductivity together with the electric field stimulation protocol. And we have also done the gene expression analysis in all these cases just to show that how specific genes are upregulated. Just to give an example, in a recent work, cases applied matrix interfaces, we have shown these rolled PVDF multi-wall carbon nanotube substrates. When you grow the stem cells, we have seen that in one case it shows to the neuron-like cells, another case it shows more like a glial-like cells. So, and that has been confirmed by electrophysiology analysis in collaboration with neuroscientists at NIMHANS, Bangalore. Now, experiments are all good, but we have to also develop theoretical understanding. So 2 things we have done. One is the biophysical basis to understand the biophysical basis for sense functionality changes. This is simple pen pencil calculations solved in the Maxwell's equations and trying to understand that what is the stress level intracellular region, what is the stress distribution extracellular region, and how the stresses also are generated, particularly the shear stresses of the cell membrane itself. And what you see here, there is significant amount of cytoskeletal tension, which leads to mechanotransduction. And this essentially facilitates a stress-driven signal transduction. Now, the question remains, when you give this electric field, what would happen to the proteins which are adsorbed on the biometric substrate and whether protein gets denatured? So to address this, we have done molecular dynamics simulation study in collaboration with Probal Maithi's group at Physics Department IISc, and we have calculated that what is the electrostatic interaction energy, and because molecular MD simulation study is typically restricted up to 100 nanoseconds, for longer time period it needs really extensive computational power. So in this particular MD domain, we have also found out that what is the RGD loop orientation and how it changes with changing in the electric field strength. And then we have found there is not much changes in the secondary structure of this fibronectin protein that we have considered in our MD simulation study. Now, in line with this cell functionality modulation, we are also curious to know whether this electric field can be used to restrict the biofilm formation. So these are some of the studies that we have recently, but we have done for quite some time, for last 10 years or so. So this is hydroxyl zinc oxide substrate on which Staphylococcus aureus bacteria is grown. Then once we give this very low intensity electric field, like 1 volt per centimeter DC electric field, we have shown that that essentially causes the membrane depolarization, leading to destruction of the biofilm. And this is one of the work that was published in Journal of Biomedical Materials Part B. Very recent work with in collaboration with endodontists, we have used this magnetic field stimulation and trying to understand that whether magnetic field stimulation can be used as a bacteriostatic stimulus to kill that Enterococcus faecalis, that is the strain which is implicated in the root canal therapy. So Enterococcus faecalis strain is very difficult to be killed under normal circumstances like using antibiotics and so on. So we have shown that up to the pulse magnetic field of 4 Tesla, there is a significant destruction of this Enterococcus faecalis strain, which is just published in International Endodontic Journal. This is one of the clinical study that we have completed in one hospital, and it is under progress in another hospital, Datta Institute of Medical Sciences in Wardha. So what we have done, we have taken the patient CT scan data and used that several software like 3D Slicer and Maze Mixer of the DICOM files just to delineate the decompressive craniectomy defect. So these kind of defects that can be a result of the traumatic brain injury, or if there is some tumor in the brain which is to be removed and that shows a lot of depression in the brain, and there also these kind of defects are created. Now, in order to retain this cranium symmetry, for example, if you see the two patients, this is a large part of the skull is being removed, and then bone flap and that place. And then we have done this study on 10 patients in the pilot study. We have found the Glasgow outcome score is around more than 4, and cranial index of symmetry that is based on analysis that is done on the basis of the analysis of the preoperative CT scan and post-operative CT scan, just to show how things on this silicon carboxylic proteins. This
Professor Basu, we are not able to hear.
Hello?
Professor Basu?
It is not audible.
Yeah. Let me call him.
Okay.
Professor Basu?
Yeah.
Yeah, so now, yeah, it has been cut.
That's fine.
Am I audible now?
Yes.
Yes, sir. Yes, sir.
Yeah, so this is the last application that I'm going to show, that is for urological reconstruction applications. So if a patient has a bladder carcinoma and let's say stage 2 and beyond, so what happens in many cases that this bladder has to be removed and urological neobladder has to be placed. Now this urological neobladder has a specific property requirement in terms of its strength, in terms of its incrustation resistance, like resistance against salt deposition and so on. Now, this kind of very wide range of property requirements cannot be made by single material. So we have developed an in situ cross-linked thermoplastic urethane polydimethyl siloxane TPU PDMS blends. And these particular blends has clinically acceptable mechanical properties in terms of the elastic stiffness, which is fairly close to that of the natural tissues, and also suture strength. Suture strength is very important. Now, subsequently, when you have we have modified that blend surface by different polycations. Then what we have found, these really does not support the bacterial growth. So essentially, it gives a lot of antibacterial properties in these particular materials. And we are currently studying the incrustation resistance in human urine, and our clinical collaborator is Fortis Memorial Research Institute in New Delhi. This brings me to the last part of my presentation, where I have promised you to show you some of my recommendations for the policymakers. So these recommendations are six-fold recommendations, and I'll go one by one. So first one is the strengthen basic research. Now, artificial and machine learning is coming up in a big way for the healthcare, but certainly its applications to the biomaterials and tissue engineering area is much limited, more so in India. Then structure property correlation biometers. Here you need high resolution multi-scale measurements, 3D bioprinting in microgravity. That is one of the ambitious program that is being launched by NASA as well as European Space Agency, but ISRO is also thinking in that line. Co-culture. We normally do culture of the single cell type, but you know that in the body, when a tissue develops, that multiple cell types also need to be investigated, and lab on chip approach.
Yeah, Professor Basu.
The cytocompat.
Professor Basu, 5 more minutes.
Yeah, I'm finishing.
Please, yes.
Yeah, so then in the year 5 and year 10, that strategic innovation centers for biometers and implants and 3D tissue manufacturing biomechanics evaluations needs to be developed. Now, in terms of the clinical context given, translational.
Somnath, please mute yourself. Somnath Roy. Yes, sir. Please continue, Professor Basu.
Yeah. In terms of the translational research, so what we believe that India should have more strategic research programs to address most prevalent human diseases in Indian context. Then GLP compliant biocompatibility testing. This is something available in CCHITRA, and CDSCO also accepts their study report because they follow that ISO standards. Then GMP regulated manufacturing facility. Many companies, they may not be having, only few companies they have 3D bioprinting of in vitro disease models and so on. Other things that which restrict that large-scale translational research is that many IVRA like institutes like Indian Veterinary Research Institutes, they do not have large animal facility, and often some of the biomaterials, some of the targeted clinical applications requires biomaterial to be tested in the pig model or in the sheep model. And these kind of animal models are often not available in many of the veterinary research institutes. So that is also very important. Industry collaborations is some of the weakest link in Indian academia, and therefore we need to involve industries more and more aggressively in our research programs in biomaterials, so that the materials that we develop, that should not be restricted in publications, that should go beyond publications. Strengthen innovation ecosystem in startups and MSMEs, implement Industry 4.0, that is very state-of-the-art and most contemporary in biomedical manufacturing centers. Fifth one is a revamped medical education. Our undergraduate medical education in MBBS degree, the students are not taught beyond that, traditional biochemistry, anatomy, physiology, and so on. They are not exposed to any of the bioengineering topics. Neither they have any exposure to their lab facilities in the field of bioengineering. So that is something to be done very soon. Science has recently finalized the establishment of the Postgraduate Medical Institute in ISC to facilitate some of the more clinically driven research, as well as to involve more clinician scientists at Indian Institute of Science. And also this kind of ecosystem is changing. In IIT Kharagpur, they have started 500-bed hospitals. IIT Kanpur, they are thinking also in that line. And some of the good changes that has happened in engineering education, like for example, in BTech program of IIT Kanpur, they have introduction to biology as a taught courses. And this introduction to biology is common to anybody who does computer science or electrical engineering or material science or chemical engineering. So this is something that allows students to think in the line to solve some of the biologically and clinically relevant research programs later on. Introduce and implement national policies and action plans, like launch national mission of bioimplants, like regulatory endorsement and global recognition of indigenous implants. That is something very important, because for Indians, anything foreign means it is good, and anything Indian means that they question the quality. So that kind of mindset has to change, and that will change only when some of the high quality biomaterials and implants will slowly penetrate into the market. And we have to implement healthcare related recommendations of NITI Aayog, that is our Government National Planning Commission, and establish regional centers of excellence for translational research. So far, the biomaterials field is being research-wise sponsored by DST-DBT, that comes under Ministry of Science and Technology, and the DCGI, the Drug Control of India, that comes under Health and Family Welfare. And unfortunately, many of the biomaterials are still treated as drugs, which is not the same. So there should be a separate committee, and there should be a separate policy level decision, like where all the related ministries should come together, and they essentially form what we call this bioimplants mission. That will help accelerate the translation and commercialization, and that will also take the innovation from academia to quickly reach the market. I'm going to acknowledge many of my academic collaborators from different national labs and academia, as well as the hospitals. And these are some of the translational research collaborators, different hospitals and companies that I'm working with, our group is working with for last several years. I'd like to acknowledge several sponsors, both governmental funding agency as well as funding agencies. And this is the dedicated group of researchers who worked and are still working day and night at my group. And it is because of them what I'm going to show you some of the research results today. And this particular group of students are, many of them, they are former alumni, and they are currently working as faculty members in some of the IITs or scientists in national labs like CGCRI and various other national labs and so on. So thank you very much.
Okay, Professor Basu, thank you so much for your very nice, illustrative, and very informative presentations in the areas of the biomedical science and engineering. It's a very good thought process you have applied in your deliberations, and covering all the global scenario as well as the national scenario, and exactly keeping those in mind where we can take our biomedical sciences, biomedical products, biomedical glass, biomedical composites, biomedical ceramics to such a position where we can have an indigenous healthy mission, a healthy India mission program. We must come out with this type of solution. And also you have given a long deliberations how science can lead to a prototype, to a product development, to a prototype development, which finds its commercialized application in many areas, for which the many startups, MSME is ready to take up the technologies in different forms, in different options. Because of the time constraint, I am sorry that I am not encouraging the students, the audience to, I know that they have many queries on these hot areas, and that to a man to like a Professor Basu. But since he's a very close to us, he's very known to us, so definitely we can approach at any point of time through emails, through phone calls, and other things are there. But I'm extremely sorry to not to put any questions to him. So once again, on behalf of the organizing committee, on behalf of CSIR-CGCRI and Director CGCRI, I sincerely thank you so much, and I remain very grateful for giving a very invaluable time, your invaluable time with us in presenting this very hot and informative topic in this today's webinar. I thank Dr. Jui for coming forward to these directions where we have a person like Professor Basu. Dr. Jui, we can go to the next speaker.
Yes, sir, we can go. But before going, I think I'll take the privilege of asking Dr. Basu, because he has suggested so many recommendations. There is only one thing which I had to ask, because we are developing biomaterials in nationwide, and then in CSIR labs, it is our mandate to develop technologies and products. But the main constraint comes with the regulatories and maybe the preclinical, which should be done in the GLP certified labs. Most of the, you already have mentioned that most of the veterinary institutes, they are not GLP certified. And wherever it is GLP certified, it incurs a very heavy cost. So, will there be changes in the policies, sir? Little bit, so that it is of help to us. Hello.
Yeah, so that's a good question, but the point is that large animal study requires a lot more strict ethical committee approval. So many of the institute ethical committee, they are essentially given the mandate for more like small animal model. For example, ISC has a Center for Animal Facility. There we can do only small animals, and Center for Neurosciences, they use a lot of monkeys. So whether it is invasive treatment or whether it's a non-invasive treatment, that also matters in terms of the ethical committee approval. So IVRI, like institutes, should change their mandate. Now, this has something to do with this top level kind of policy changes. So once they have this more like large animal model, currently you can do large animal study only in Suchitra. They have, I think, but that is also limited. There is a lot of waiting time and so on. So in a country like India, it is not sufficient to have only one institute having this kind of facilities. IVRI, which is there at Ijjat Nagar in UP, Uttar Pradesh, I think they also have some of the large animal, but many of the other IVRI they do not have.
Okay, sir, thank you. And yes, the politicians always keep on saying of Make in India mission, but there needs to be change in, the intermittent arrangements and the policies to facilitate our work. To go forward with the products in a more easy way, sir. That is the understanding. I mean, all of us have. We face a lot of constraint, as we have already mentioned. Thank you, sir.
Thank you.
Yeah. Thank you, sir. Thank you very much for your
Sir,
time.
sir, will it be possible, I mean, once more to put the video on and we take a snap, whoever is present in the audience? And madam, I think she's, yes. Yes, sir. And all the other participants, please make your videos on. I wanted to take a snap along with sir. And madam actually is not visible.
Yeah, turn on.
Ognivo is taking.
Yes, I have taken a snap and I have also shared it with you in the email. So
Okay.
kindly check. Yeah.
Oh, please, please make all the videos on because I'm not able to see everybody. Yes, sir. Thank you.
Hello, I am not. I don't know if there is something problem. Thank you, Vikram. It was a very nice lecture.
Thank